Hi everyone, I'm Cheng from National Tsinghua University. I'm very excited to share with you our research about branch actin filament forms size-dependent diffusion barrier around centrosome, which is just published in EMBO report. In this study, we ask, as microtubule organizing center, platform for cellulogenesis, and signaling hub, how do centrosomes maintain their microenvironment without membrane in the crowded and busy cytoplasm? To understand this, in the following experiment, we investigated centrosome at 3 subcompartments, from core to the exterior region, centriolar lumen, pericentriolar matrix, and centriolar satellites. On top of it, we incorporated chemically inducible dimerization system in which FRB and FKBP can be joined together upon rapamycin treatment. We conjugate FRB with region of interest and FKBP with protein of interest. Such dimerization allows us to see translocation of probe to targeting area when they are in close proximity. Primarily, we established a centrosome CID. We associate FRB with centrosome marker and FKBP with probes. Theoretically, without diffusion barrier, we will see probe signal accumulation at targeted area upon rapamycin treatment. On the other hand, presence of impervious diffusion barrier can hinder the access of diffusive probe, and therefore we will see no signal accumulation. Ideally, we want the trapping observed here is due to probe diffusion, not other transport mechanism. Therefore, we use FREP assay to select centrosome targeting protein with low kinetics. This is the location of 3 selected centrosome targeting protein. Centrin 2 is centriolar lumen, CDK5 Rep4 domain at pericentriolar matrix, and PCM1F2 at centriolar satellites. We recruited probes ranging from stoke radius 3.2 to 7.6 nanometer to each site. I will abbreviate these probes by their size, calling them probe 3.2 or probe 4.1. First of all probes to centriolar satellites. Blue fluorescents are centriolar satellites. Green fluorescents are probes from small to large. Probes rapidly translocate within half a minute post-rapamycin treatment. And then all probes to pericentriolar matrix. Blue fluorescent is packed-tech pericentriolar matrix. Interestingly, the larger the probes, the slower the translocation. It is now already less likely to see accumulation of too bulky probe, indicating obstructed diffusion. Similarly, all probes to centriolar lumens. Blue fluorescent is centrin-tech centriolar lumen. Consistently, bulky probes show slower or no translocation. At this point, we argue that 3-minute post-rapamycin treatment was perhaps not enough for visible accumulation for bulky probes. So we extended the time and recorded translocation 2 hours post-rapamycin treatment. Coherently, translocation probability of bulky probe was still low at the same locales. Therefore, we infer that the barrier lies outside pericentriolar matrix. We then try to investigate the composition of this barrier. We postulate that microtubule and actin, the 2 rich elements around centrosome, may be the major component. Hypothetically, removal of the major component would significantly change the permeability pattern. To start, we use non-cadazole treatment to disrupt microtubule. The diffusion pattern remained unchanged. Following, we disrupt actin with ARC2/3 inhibitor CK666 and actin polymerization inhibitor electronicling A, respectively, to examine actin's role. The blue fluorescent here are pericentriolar matrix and lumen. Green fluorescence is probe 6.3. Before actin disruption, the barrier is not permeable to the probe. It was after actin disruption that the probe can move across the barrier. We take a look at different probes. X is probe size, Y is translocation probability. The data suggests that actin disruption had allowed bulky probe to diffuse across the barrier. Such change in the diffusion pattern suggests that actin filament forms centrosome diffusion barrier. Since actin disruption in cell is dynamic, we're also curious whether rising actin level decrease barrier permeability. Therefore, we took advantage of the temporary increase of actin filament around centrosome during anaphase and synchronized our cell for the following experiments. We collected interphase and anaphase cell. Pericentriolar matrix is in blue, probe 4.5 in green, and chromatin in red. Usually, probe 4.5 is small enough to diffuse across the barrier. The video demonstrates that diffusion was obstructed during anaphase. The same effect can also be observed at probe 6.3. The results suggest that permeability of diffusion barrier do drop when actin level increase. And we can rescue the effect by using CK666 to perturb the anaphase actin filament, and the same probe can move across the barrier again. And this is corresponding to our previous data. We have come to infer that permeability of centrosome diffusion barrier is tightly regulated by branch actin filament, and now we want to know the role of such regulation to microtubule organizing center. Therefore, we investigated the change in microtubule after disrupting the barrier. Here we mark centrosomes red, and component of microtubule nucleators, the γ-tubule green. Image and quantitative data indicates that disrupting diffusion barrier significantly increased γ-tubulin level at centrosome. Although γ-tubulin was rather small in size, the observation encourages us to run further examination using GCP4 protein, a larger component in γ-TuRC. We examined GCP4 dynamics using FREP assay. As the video shows, disrupting diffusion barrier increased GCP4 shelling rate at centrosome. Quantitative data suggests significant increase in mobile fraction and shorter recovery half-time after barrier disruption. We tracked microtubule using EB1 and observed the nascent microtubule density at centrosome over a period of time. You can see the increase in the number of nascent microtubule at centrosome with disrupted diffusion barrier. In conclusion, our study demonstrates that the permeability of the diffusion barrier between centriolar satellites and pericentriolar matrix is tightly regulated by branch actin filament. The barrier gates microtubule nucleation by interfering with γ-TuRC recruitment. And that's all for my presentation. Thank you for your attention.